Hello guys, today we'll be discussing the reprogramming the somatic cells to pluripotency. This is our topic. So start off with somatic cells. What are somatic cells? Basically, somatic cells are differentiated cells which don't undergo under differentiation. They do not have the ability to differentiate into other cell types. For example, it could be neuron, epithelial cell, blood cells, or muscle cells. These are some of the example of somatic cells. So the second type of cells are pluripotent cells. So they are the cells they are capable of differentiating into almost every type of the body. This process of differentiation is called as pluripotency. The examples are embryonic stem cells, induced pluripotent stem cells, and we can see from the diagram that from in vitro fertilized egg we get blastocysts. From there we get pluripotent cells, and from that pluripotent cells we get different type of cells including blood cells, cardiac cells, and neural cells. Moving towards the next slide, here what we have, how it all started, how the topic, what is the reprogression of reprogression, what is of somatic cells to reprogramming of somatic cells to to pluripotent cells started. So initially in 1981, the stem cells were extracted from mouse, and in 1998, stem cells were extracted from humans. So from that, it started, and after a long time, like just 8 years, and then Shinya Yamanaka, a scientist, she used to convert some somatic human cells into IPCs, that is induced pluripotent cells, that are basically stem cells. So she used OCT4, SOX2, and KIF4, but later on it was invented that chemicals and electrons can also induce induction of IPCs from somatic cells. So why do we need to take reprogramming somatic cell development takes place? Why do we need to take reprogramming somatic cells? Why we need to this need of this reprogramming? So basically, we are asking, why do we need this reprogramming? Because as you have learned from every biologist knew about stem cell research and application, so we have to know about the stem cell and application. That's why we want to undergo reprogramming of somatic cells. Attempt to address the ethical concern regarding the use of embryonic stem cells. So it will be discussed in the upcoming slide. The third one is understanding human development and cellular programming technologies, reprogramming technologies. And the fourth one is patient-specific treatments that basically include regenerative medicines, and it also became the foundation for regenerative medicines. So the second point will be discussed here. So why can we use the pluripotent stem directly? So the first one is ethical concern, risk of tumorigenesis, and third one is immune rejection. So the ethical concern include extraction of embryo causes destruction of embryos. So it is basically an ethical concern because the embryo cells dies after the extraction process. The risk of tumorigenesis. The second one is the capacity of pluripotent cell to form tetramas, a type of tumor that contain variety of tissue types. It is also a risk. That's why we don't use pluripotent stem cell directly. And the third one is immune rejection. The therapies based on stem cells, they may suffer from immune rejection also. There is a high chance that people will get immune rejection by using the directly pluripotent cells. So that's all from today.